Hi, first off, I'd like to just say that I'm very grateful to be receiving this award, and I'm very excited for the future work that this will help enable in our lab. I'm currently an assistant professor here at UC San Diego in the Departments of Medicine and Computer Science. It's been about 4 years or so since we've opened our lab, and we've been really interested in understanding repetitive regions of the genome, which are often ignored in most genetic studies. And we've been particularly interested in how repetitive regions of the genome might be contributing to psychiatric disorders such as schizophrenia and autism. So it's now very well established that most psychiatric disorders have a very strong genetic component. And this slide is presenting data from a study in 1991 looking at someone's risk to have schizophrenia based on their relationship to a schizophrenic individuals. So for example, if someone has a first cousin that has schizophrenia, they have only moderately increased risk to have schizophrenia themselves over the 1% or so baseline in the population. However, if your siblings or your parents have schizophrenia, then that risk goes up to 5 to 10%. If someone's identical twin has schizophrenia, then they have almost 50% chance to have schizophrenia risk themselves. And really this risk goes with the fraction of the genome shared. So the higher the percentage of the genome that you share with a schizophrenic individual, the higher risk you would have of having schizophrenia yourself. And of course, most psychiatric disorders, as well as other types of disorders, are due to a combination of genetic or environmental effects. But at least for schizophrenia and autism, it's been consistently estimated that about 80% of the variation and risk for these conditions across individuals is actually due to genetic factors. But another thing that has now become very clear is that the genetics of most psychiatric disorders is very complex. Unlike our traditional view of genetic disorders where we think about a mutation in one gene leading to one disorder, for both schizophrenia and autism, there are actually many, many different genes across the genome that could be contributing to risk for those conditions. And this makes it actually quite a lot more difficult to study from a genetic standpoint, because you might have 100 different autism patients and we'll be looking at the genomes of those patients, but there might be 100 different genetic causes, a different cause for each of those different individuals. So really to get a handle on the genetics of these types of disorders, we need to have really huge sample sizes collected to start robustly implicating even a single gene. And over the past decade or so, there's been a lot of progress made in identifying many genes that are now robustly linked with both schizophrenia and autism. And that's really only been made possible by these huge international collaborations that have collected increasingly larger sets of patients in order to get a handle on the genetics of these disorders. And I think this is really telling if we just look at the history of schizophrenia genetics research over the last 10 years or so. The first genome-wide, or one of the first genome-wide studies of schizophrenia done back in 2009 had collected about 3,500 patients with schizophrenia and compared their genomes to control genomes. And even with that seemingly really large sample size, no single gene was able to be reliably implicated in schizophrenia. A couple years later, that sample size rose up to 10,000, and there the first couple of genes began to emerge where we could robustly say mutations in those genes is linked to schizophrenia. But only after getting to around 37,000 cases in 2014, a major landmark study was able to implicate more than 100 different genes across the genome in schizophrenia. And as sample sizes have continued to increase through these collaborative efforts, that number of genes just continues to go up. And if you look at autism, we see a similar story starting to emerge where there's many different genes that are implicated in autism, but only by collecting these huge cohorts consisting of many thousands of individuals can that picture really start to emerge. And this list of genes just keeps on growing as those sample sizes keep growing. But maybe one other takeaway that we've seen from this is despite the fact that these studies have been able to implicate all of these genes, we know this is just the tip of the iceberg and it's not telling the whole story. Mutations in these genes still explain a minority of cases of psychiatric disease. So if a patient walks into the clinic, only for a minority of cases could we point to, say, a mutation in one of these genes that has been identified and say, no, that is the cause of the condition in this specific patient. So there's still really a lot that is missing from the picture. Almost all of the studies that were referenced on the previous slide have been looking at really one particular type of way that you can have variations in DNA that could lead to disease. And that is looking at these small changes in DNA between individuals that we could really think about as being kind of like spelling changes. So for example, there might be a position in the genome where almost everybody in the population has a T nucleotide at that position, but then occasionally you see a different nucleotide, say an A at that position. So just really small spelling changes in the DNA where there's just a substitution where most people have one base and some have a different base or nucleotide at that position. Sometimes we refer to these really small spelling changes as single nucleotide polymorphisms or SNPs. So I might use that term later in the presentation. So these SNPs are really just referring to these small spelling changes. So these SNPs are really widespread and a large source of genetic variation between different people and their DNA. But these SNPs are really just part of the story and how genomes can vary from one another. And if you go and look closely at the DNA sequence of the human genome, you'll find a lot of places that don't really look like the variation of those SNPs that we saw on the previous slide. And a lot of places in the genome, you'll see places that look like this example shown here, where you have these repetitive regions where maybe there's a short sequence of DNA, like the CAG example shown in this slide, that is repeated many times one after another. Right? Because these regions are repetitive, it actually causes the machinery that replicates DNA when our cells divide to occasionally get confused and make mistakes. So you can get actually a lot of variation that will either add or delete copies of this repeat. So if you were to compare different copies of the human genome between different people, you might see variation in terms of the number of copies of this short repeat sequence. Right? And here I showed a CAG repeat as an example, but it doesn't have to be a CAG repeat. It could be another short nucleotide sequence such as AC or CCG. We'll see a couple of other examples later on. These repetitive regions of the genome we refer to as short tandem repeats, or STRs, and so I'll use that term later on to refer to these repetitive regions. So why are we so interested in studying these repetitive regions of the genome? Well, one of the reasons is that these are just so widespread throughout the human genome. If you look at a single individual's genome, you might find more than 1 million of these STRs scattered throughout the human genome. If you were to just zoom in on one of those STRs in particular and just look at the number of repeats that you see in different people across the population, you'll see that often these are extremely variable across different people. So if you were to look at, for example, a single CAG repeat, maybe you would have 6 copies of that repeat in your genome, and your brother would have 5 copies, and your friend might have 10 copies of CAG repeat. So a lot of variation in the number of copies that you have. And you'll note that for each of these people, I showed 2 copies of the repeat. That's just because for most regions of the genome, you have 2 copies, one from mom and one from dad. So at most of these STRs, you would have 2 different copy numbers present. But if you were to just take all of these copies across all the individuals in the population and look at a distribution, often you see this huge spread, this huge amount of variation just at this single place in the genome. If we were looking at SNPs, all we'd be working with is someone has an A versus a T. But if we're looking at these STRs, we have this huge spectrum of variation where there's just a big range of different numbers of copies of the repeat. This is just at a single repeat. You can see a similar story when you look at the rest of those 1 million or so STRs across the genome. So that's the second reason that we're really interested in these repetitive regions, is just that they're so variable across the population. In fact, they're one of the biggest sources of genetic variation between the genomes of different humans that we know about. And of course, a third reason that we're really interested in these repetitive regions is because it can be associated with many different human traits or diseases. And we already know of several dozen or so of these STRs that can be implicated in disease. I've shown a couple of examples here on this slide. On the left is showing an example STR that is implicated in Fragile X syndrome, which actually accounts for about 5% of cases of autism spectrum disorders. In this example, you have one of these STRs, in this case it's a CGG repeat, right upstream of a gene called FMR1. Most people would have about 30 copies of CGG in their genomes, but in some cases you can get this repeat expanding. If it keeps on expanding and gets to more than 200 copies of CGG, then that ends up causing Fragile X syndrome, which is a form of mental retardation, and again, it's linked to autism spectrum disorders. Another well-known example is Huntington's disease. In this case, this is another gene, HTT or Huntington. You have a CAG repeat within this gene. Most people have around 20 to 30 copies. If that expands to more than 40 copies, you get Huntington's disease, which is a devastating neurological disorder. Interestingly, almost all of the repeat expansion disorders that we know about actually end up causing some kind of a neuropsychiatric phenotype. So we know about many of these example STRs that are involved in disease, but we mentioned a couple of slides ago that there's actually 1 million or so of these types of repeats or STRs across our genome. Most of these have not been well studied, as most recent studies have focused on these SNP variants or these spelling mistakes, which are far easier to analyze. So our major question has been, well, what can we learn by looking at the rest of these 1 million or so STRs across the genome? And if we can start integrating those into these genome-wide studies, can we help explain some of that missing genetic component of psychiatric disorders that was missed when we only looked at these spelling mistakes or these SNPs? So that's really a major goal of our lab, is both to develop and apply frameworks to analyze these STRs or these repeats at population scale in order to learn about how they're affecting psychiatric and other traits. And there are two kind of main frameworks that we've been using to try and get at this. And the first type of framework, we've been analyzing STR mutations in families, where maybe we have a family where there's a single individual affected by something like autism spectrum disorders. We can look for changes in the DNA at these STRs that is specific to the individual that is affected, but not found in the other members of the family. And the second type of framework we've been looking at is more of a population level analysis, where we can compare the genomes at these STRs and sets of patients that are affected by disorder and compare those to the genomes of people that are unaffected by that disorder. And I'll just tell you two kind of case studies that we've been looking at here. And the first doing these sort of family studies in autism spectrum disorders, and the second doing this population level analysis in schizophrenia. So one of the main projects in the lab has been looking at mutations in these repeats and how those could be contributing to autism spectrum disorders. And for that, we're relying on this really, really great data set generated by the Simons Foundation called the Simon Simplex Collection, where we have whole genome sequencing for about 2,000 families that have this sort of quad structure, where we have the two parents, one child unaffected by autism and one child affected by autism. And this data set, we've been focusing on something called de novo mutations, where we see a variant at a repeat that is present only in the child that was affected by autism, but that variant is not found in any other family members, which is really an indication that that variant kind of arose by a mutation, maybe in the sperm or the egg cell for that child, because that variant is not present in the DNA of either of the parents. And we've had a couple of major goals in this project. One is just to be able to develop the computational tools we need to analyze STRs from this type of data. We'd like to answer some basic questions, like how many mutations at STRs does each child typically have? And can we determine specific mutations in these STRs that might be actually leading to the autism in the affected children? So a lot of our time has been spent figuring out a lot of the computational challenges that we have to deal with. So the data that we get out from the sequencing of these individuals after we perform Illumina whole genome sequencing looks something like this, where we just have a collection of nucleotides from these samples, and we have literally millions or billions of nucleotide sequences. And we have to figure out how do we go from this bunch of letters to actually inferring if there were any mutations that occurred in the child. So we spent several years developing a pipeline that can take this collection of nucleotides we get off the sequencer, figure out at each repeat how many copies each person has in the family, and then trying to integrate that information to figure out the probability that a mutation might have occurred at each child, at each STR across the genome. And this is working reasonably well, but sequencing data is still noisy and prone to technical errors, so we also have to do a lot of work to go and kind of validate some of these findings back in the wet lab. Here's just showing one example of how we can go back and validate one of these mutations. This is an example family where we had a CGG repeat in this gene reelin, known to be implicated in autism. We saw that the child had 12 copies of that repeat, where the parents had either 8 or 10 copies. So it looks like there was a mutation in this child. Well, we can go in the lab and apply a different technique where we just analyze the size of the fragment around this repeat. And indeed, we were able to find that there's a band here that was only specifically found in the affected child, indicating that this indeed was a mutation. This wasn't found in either of the other family members here. Once we have this pipeline set up, we can start to try and answer some basic questions, like how many of these mutations do we see in a single genome? Overall, we found that either in healthy or affected individuals, we see between 50 and 100 of these STR mutations per child, which is about as many as could be found by these SNP-based studies. So we're really kind of doubling the number of these types of mutations we have to look at in each genome that we analyze. We also found some interesting trends. Like other types of mutations, about 3/4 of these mutations are probably coming from the father. The number of mutations in the child increases as a function of the age of the father, probably because the sperm cells in fathers are continually dividing throughout their lifetime, so older fathers tend to have more mutations passed on to the children. But we also found interesting things about mutations that come from mom. Even though you get more mutations from dad, it turned out the mutations that you get from mom tend to be bigger. So most mutations from the mother would result in changes of the repeat copy number that are greater than 1, whereas almost all the mutations that we saw from dad were just making minor changes to the copy number, so adding or decreasing a single repeat copy number whenever there actually was a mutation. So more mutations from dad, but maybe more severe mutations from mom. And finally, before going and looking at the autism-affected children, given that we see so many of these mutations, even in healthy individuals, a major challenge that we had to figure out is how can we actually figure out, for a particular mutation, is that mutation just kind of neutral and not really doing anything, or is that mutation itself actually contributing to autism in that child? So I have a really talented undergrad student in the lab, Bonnie Huang, who has developed a method called SISTER that integrates both information about each of these repeats in healthy populations, but also some kind of detailed mathematical models of how these things mutate in order to allow us to develop a score that can predict for a particular mutation, do we expect that that mutation is going to have a severe impact on fitness or disease, or is that mutation kind of just neutral and not really doing anything? So now we've gone and we can apply these techniques to ask, do we see differences in terms of these STR mutations in the children that are affected by autism compared to their unaffected siblings? And the main metric that we've used to analyze this is a simple score called the relative risk, where we're simply just counting the number of mutations we see in the affected child and comparing that to the number of mutations we see in unaffected siblings. If we look at genome-wide at these STR mutations, we actually do see a modest overall increase in the number of these mutations in the affected child compared to the unaffected children. So this relative risk is greater than 1. and it's actually highly statistically significant, but still quite modest. They have very similar numbers, but a slight increase in the affected children. But then we said, well, most of these mutations we know are probably neutral, and maybe if we restrict to the set of the mutations that are most interesting, maybe this difference will be increased between the affected child compared to their unaffected siblings. And indeed, if we restrict our analysis only to STRs that are found in or near genes that are expressed in fetal brain, this relative risk starts to go up. But then when we apply these sister scores to restrict only to STR mutations that are predicted to have the most severe effect, that's where we get the highest relative risk here of 2.5. So we see 2.5 times as many of these predicted severe mutations in the affected versus the unaffected siblings. Interestingly, this analysis has brought up mutations of these STRs and genes that we already knew were implicated in autism, but it's also brought up a lot of interesting novel candidate genes that weren't implicated by previous analyses that were only looking at these spelling changes or these SNPs that we're now following up. I'll just end by briefly telling you about this second type of analysis, this population level analysis that we've been applying to understand the role of these repeats or these STRs in schizophrenia. In this setting, where we're doing a little bit different, instead of families, we have sets of unrelated individuals, some of them that are affected and others unaffected by schizophrenia. Here we're looking at a really large data set collected by the PGC, or the Psychiatric Genomics Consortium, where we have almost 27,000 cases that have been collected through this international effort. And here we're doing something conceptually very simple. We're just looking at each repeat in the genome. We would like to test if the length of that repeat is on average different between the affected versus the unaffected patients in our cohort. And we can repeat this type of analysis for the million or so STRs across the genome. This sounds conceptually very simple. There are many technical challenges that we've had to overcome in order to get this to work. One of the challenges is that most of these large studies don't actually observe the repeat regions. Turned out to be a lot cheaper just to look at a really small subset of SNPs across the genome to perform these studies. So we don't actually observe the STR sequence for each sample. So Shubham Saini, a graduate student in the lab, has developed a framework that allows us to basically guesstimate the number of repeats for each person based on patterns that we've previously observed in other data sets. And it turns out we can guesstimate the number or impute the number of repeats in each sample quite well. On average, 97% of our guesses appear to be correct when we're imputing this number of repeat copy numbers. So now we can use those estimates of repeat copy number in each person to do this analysis, to compare the number of copies of the repeat at each STR in the genome between the cases and controls. When we do this analysis, we end up with these cool plots called Manhattan plots, where here we're looking just across the genome. Each dot here represents a single STR that we tested, and the height of the dot is representative of how strongly that STR was associated with schizophrenia risk. And it's called a Manhattan plot because you see these skyscrapers pointing up that are really pointing out the regions of the genome that are most important. Similar to our autism study, a lot of these kind of skyscrapers are picking out regions of the genome that had already been implicated by SNP studies. But we also find several dozen novel hits where we see regions of the genome where you have a repeat associated with schizophrenia risk that wasn't previously identified by these SNP-based studies. Here's just one example of the type of thing that we find. In this example, there's an STR that is just a bunch of repeats of T in a row. And the more T's a person has in their genome, the higher their risk of schizophrenia. And this is a novel candidate gene, DSN, that hadn't been implicated by previous SNP-based studies. So just to conclude, we're working hard on developing and applying these methods to look at these STRs at genome scale and population scale to learn about their role in psychiatric disorders. This has uncovered many specific STRs that show strong association with either autism or schizophrenia. And now kind of the next steps is going to be figuring out actually biologically how are changes in these repeats resulting in changes that eventually lead to disease. Because only after we understand the biological mechanisms can we make progress towards treatment and understanding of the biology here. And I guess another main direction that we'd love to think about in the future is how to incorporate more diverse population groups in these analyses. Most of these have been based on individuals of European descent, and it's become clear that by incorporating more diverse groups into these type of analyses, we can identify more and more regions of the genome that are important to larger numbers of people. And finally, I'd just like to end by thanking all of the great members of my lab that have really done most of the work that is presented here, and also our strong collaborators and our funding sources from NIH and the Simons Foundation that have enabled us to do this work.